Good afternoon, Conference Chair and distinguished guests. Thank you for giving us the opportunity to present today. Dr. Kishwar Ali and myself are part of a team of researchers here at the College of the North Atlantic Qatar campus, specializing in applied sciences and technology. Today's topic is on spatial temporal changes in Qatar in the wake of global climate change, with special reference to the floristic diversity of the country. This was a multifaceted study designed to assess the conservation status of Qatar's endemic flora by determining its floristic distribution and significance using modern GIS, or geographical information systems, maxent niche distribution modeling techniques, and phytochemical extraction and analyses. We've applied GIS, maxent niche modeling, and phytochemical analysis to get the required results that we obtained. For spatial and temporal mapping and analysis of the vegetation and classification analysis of land use changes, we applied GIS techniques, and ArcMap 10.8.1 was used to obtain unsupervised classification of Landsat 8 images to obtain the various land use classes. For phytochemical extraction, we use standard ethanol methodology to obtain extracts used in the microbial trials. Dry samples of ground plants were analyzed for phytochemical identification. Land use classification was determined using Landsat 8 images using ArcMap 10.8.1, and you can see that scarce vegetation is evident from these maps, and urban development continues to increase. From the map A, we see the urban development on the east and the west coasts of Qatar with scarce vegetation down through the middle. On map B, we notice the red dots, and these are the sample sites we used when collecting and ground truthing vegetation. The maxent niche models generated show the probability distribution of the prominent indigenous plants. It was determined that some of the sample plants have specific niches as indicated in the coloration of these maps generated. Red being the highest probability, and green being the lowest probability. Here we show the difference in two species, one being a monocot and the other a succulent. Fontamesi was found to have the highest total effect with a total of 83 millimeters in zone of inhibition. And it had this effect on the greatest number of bacterial species. In this case, it was 5 species. The lowest effect was shown by Limonium auxiliaire, which had a total of 24 millimeters for the zone of influence, and it only affected 3 species of bacteria. The FTIR spectroscopic analysis of the crude plant extracts was performed on the samples, and this showed troughs of absorbance at wavelengths synonymous with those of alkaloids, flavonoids, and polyphenols. So in conclusion, Qatar is undergoing a rapid change in regards to its land use. Qatar has scarce but important and valued flora which could be exploited for phytochemical extraction and commercialization. Some of these plants have specific niches which could be affected by climate change in the future and can have an impact on the overall biodiversity of Qatar. I'll pass it over to my esteemed colleague, Dr. Kishwar Ali, who will comment on some acknowledgements and an invitation to everybody.
Thank you very much, Aaron. On the notes to on acknowledgements, I really appreciate, or the whole team really appreciate the College of North Atlantic Qatar, Qatar's administration, President Dr. Salim Naimi, and Professor Dr. Michael and his team at the research office for funding this project under the seed fund scheme. That allowed us the opportunity to work on developing our research profile in basic and applied research, and enabled us to initiate a sound foundation. I would also ask people researchers out there, that CNAQ is open for collaboration with any national and international organization. Please do contact us on the details given. We have got a number of opportunities, funding opportunities available in the form of NPRPs, Europe projects, and they are funded by Qatar National Research Fund. So please do contact us in this regard. If you got any questions, we still have got a couple of minutes. Please do ask us questions or email us, and we'll respond accordingly. Thank you very much for your time.
Thank you.